Namaskar, I am Biju Dharmapalan and you are watching Science Time, a show that brings you the latest developments from the world of science and technology. Let's first look at some of the exciting stories. Lab-grown blood transfused into humans. Egg whites can be transformed into a material capable of filtering microplastics from seawater. The Enigma of plants' compact sperm is demystified. Now let's see the news in detail. Red blood cells grown in a laboratory have now been transfused into another person in a world-first clinical trial. The manufactured blood cells were grown from stem cells from donors. The red cells were then transfused into volunteers in the RESTORE randomized controlled clinical trial. The RESTORE trial is a joint research initiative by NHS Blood and Transplant and the University of Bristol, working along with University of Cambridge and St. Thomas NHS Foundation Trust, National Institute for Healthcare Research, Cambridge, Clinical Research Facility, and Cambridge University Hospitals NHS Foundation Trust. This is the first time lab-grown blood from an allogenic donor has been transfused. So far, 2 people have been transfused with the lab-grown red cells. They were closely monitored. No untoward side effects were reported so far, and the patients who have not been named are well and healthy according to the researchers. The amount of lab-grown cells being infused varies but is around 5 to 10 milliliters or about 1 to 2 teaspoons. The trial is looking at the lifespan of the lab-grown cells compared with infusions of standard red blood cells from the same donor. Because the lab-grown blood cells are all fresh, researchers expect them to perform better than a similar transfusion of standard donated red cells, which contain cells of varying ages. If proved safe and effective, manufactured blood cells could in time revolutionize treatments for people with blood disorders such as sickle cell and rare blood types. Let's move to the next story. Researchers at Princeton Engineering have found a way to turn your breakfast food into a new material that can cheaply remove salt and microplastic from seawater. The researchers used egg whites to create an aerogel, a lightweight and porous material that can be used in many types of applications, including water filtration, energy storage, and sound and thermal insulation. Egg whites are a complex system of almost pure protein that, when freeze-dried and heated to 900 degrees Celsius in an environment without oxygen, create a structure of interconnected strands of carbon fibers and sheets of graphene. In a paper published in the journal Materials Today, researchers showed that the resulting material could remove salt and microplastic from seawater with 98% and 99% efficiency respectively. Let's move to the next story. In cells with a nucleus, or the eukaryotic cells, DNA is packaged into structures called nucleosomes by wrapping around core histone proteins named H2A, H2B, H3 and H4. The nucleosomes form DNA protein complex chromatin, thereby packing DNA into the cell's nucleus and regulating gene transcription. Animals and several ancient plant lineages hypercondense DNA in sperm cells by replacing histones with proteins called protamines. This produces a compact sperm head that increases swimming velocity. However, traits of such condensation include a nearly complete loss of histone-mediated control of gene expression and a suppression of transcription. Flowering plants do not have protamines and produce sperm cells that show active gene expression and cannot swim. It has remained unclear how sperm chromatin is condensed in flowering plants without using protamines, or how this condensation mechanism allows transcription to occur. A research team from the John Innes Center have discovered a mechanism of flowering plants, sperm compaction, and gathered clues as to why it is required. Researchers have shown how in flowering plants, sperm chromatin, a complex of DNA and protein, is compacted by a special histone protein that spontaneously self-aggregates as oil droplets do in water, a phenomenon known as phase separation. Researchers used super resolution microscopy, comparative proteomics, single cell type epigenomic sequencing, and 3D genome mapping to investigate this mystery. The research findings are published in the recent issue of the journal Nature. Now let's move to the Science Quest segment. The question for this week's Science Quest is, why do bees make honey? Honey bees make honey to store up as food to last them through the winter months. During the coldest time of year, there are fewer flowers from which to collect nectar, and honey bees are unable to forage. Thanks to their supply of honey, members of these colonies survive through the winter, unlike in most bumblebee colonies, where only the queen bee survives by hibernating underground. Honey is usually made from nectar, the sweet liquid produced by the flowers to entice bees and spread their pollen. A worker bee sucks up the nectar through a long thin tube called proboscis and keeps it in a special honey stomach known as the crop, which can hold up to 80% of bees' weight in nectar. Inside the bee's enzyme, including one called invertase, begin to break down the complex sugars into simpler ones that are less prone to crystallizing. Once the worker returns to the hive, forager bees pass the nectar to each other from mouth to mouth. Workers that are younger than the foragers then pack the nectar into hexagon-shaped cells in the honeycomb that are made of bee wax. Next, they fan the nectar with their wings to encourage evaporation. While nectar is 70 to 80% water, these processes reduce its water content to around 18%. This reduction in water turns the nectar into honey. The high concentration of sugar, meanwhile, ensures that bacteria and fungi can't grow, meaning honey can be stored indefinitely without spoiling. The honey is covered with fresh bee wax and stored in the cell until it is needed. A healthy colony can produce 2 or 3 times the amount of honey it needs. So it isn't a problem for them if humans take little bit. With this, we come to the end of this edition of Science Time. Keep sending your feedback and suggestions through email. Our email ID is indiascience@vigyanprasad.gov.in. We will be back with more exciting stories from the world of science next week. Stay tuned to India Science.